Welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, entertainment, and I'm sorry, not entertainment, that's later today. This is Big Discussions 76, science and technology. Okay, got that right. Today is November 8th. 2020. It's a Sunday morning here in the DMB. The sun is out. And I don't know who's going to watch this one. I see 3 people have come in already. I don't know who's going to watch this one. I know a lot of folks are still feeling the euphoria of the political stuff from yesterday. But I realized this week that I hadn't watered this channel in a while. With the Big Ten football season starting back, I've been uploading a lot of content over there and doing live streams over there, but I hadn't done anything over here. And so I wanted to come back and do something over here in the science space. And I have some more cool things that are coming from this channel, probably within the next week. So but this topic, I plan to only go for about an hour or so, unless some people have questions and want to come in here and ask me about it and talk about it. But the cool thing about StreamYard is no matter how many live viewers you get, people can always watch it on the playback. If this is your first time here, please like the stream, please share it, please subscribe. I think this channel is sitting right at 527 subscribers. So the next milestone is to get to 600. So the likes are free and the subscriptions are free. If you want to donate to the channel, my Cash App is below in the description box. And so with that, let's get started because I have some more to do today on one of my other channels. So this topic is this is a science topic. So I know I've done a lot of pre-records and I've had Nicole up. We've tag teamed some things, but I think this will be the first time I've kind of hit a science topic the way the Scientist Supreme does. And I hope I don't lose you all, who's ever in here. I see some more folks coming in. I hope I don't lose you. And I hope that you're able to get something out of this. One of the reasons I started this channel for a number of reasons, but one of them is STEM advocacy. I think that as a Black male, I think, well, as Black people, we need to have more conversations like this about the sciences. Society is only going to get technologically more advanced, and there are going to be a lot of careers going forward in the sciences. And with policies put in place, well, we don't know what policies will be put in place going forward, but you just want to give everyone a chance in your community to compete with everyone else from around the world. And within the country. So J3, thank you for coming. I see you here. BGS brought me here. Okay, appreciate that. So today I'm going to talk about a discussion on the science of absorption, distribution, metabolism, and excretion. Okay, so that is a subset of the 2 sciences that I've been trained in, pharmacology and toxicology. And that's a picture of me right there at my lab bench at the University of Michigan about 15 years ago. I was a younger lad, a little more innocent and bright-eyed and bushy-tailed. Right now, I work in the federal government in regulatory affairs. That's about the extent of what I normally say about where I work right now, for just to protect some privacy and those kinds of things. But it turns out that this week I've been delegated to give a training at my office regarding this particular area, absorption, distribution, metabolism, and excretion. which is a subset of pharmacology and toxicology. And it's very relevant to any disease state or any sickness a person has. This is very, very relevant. And hopefully when this is done, I will have convinced you that it is. If you have any questions, leave them in the chat. At some point, I'm going to throw the link in if anyone else wants to come up. So what I'm going to do, I hope this goes smoothly. I'm going to enable the screen share. Shout out to the Scientist Supreme, the Lioness. She showed me how to do this trick. I always shout her out for this. I'm going to do my screen share really quickly. In addition to being a scientist and a YouTube content creator, I was a writer before coming over here to YouTube. This is my blogging platform, the Big Words blog site. There's a significance to that name, but when I first launched my blogging platform, I wrote a couple of pieces on the sciences that I was trained in, and I wrote these up to distill them down and to make these areas more digestible for people from my community in particular who have had no exposure to these scientists. Because I get, when I tell people, especially here in the DMV, and there are a lot of professionals here in the DMV, keep in mind, lawyers, program analysts, career politicians, those kinds of things. When I tell them I have a background in pharmacology, they immediately think pharmacy. So I wanted to write this series up to kind of, it got a little frustrating after a while, but I wanted to write this series up so that people could go and say, this is what it is, this is what it is, this is what it's not, and also to open the door for others to follow these paths as well. So I wrote this blog post back in 2017, so it's almost 3 years old. Wow. I'm going to read this, and I'm going to use this as my guiding light during this live stream. And I'm going to show some pictures here as I go along. Hopefully I won't confuse anyone. If you have any questions, please ask. But this blog post is going to be our guide. I'm going to read through it, and I'm going to stop at certain points and show a couple of visuals, and then we'll wrap up. Hey, Nicole, thank you for coming. Thank you for stopping by for as long as you're here. So with that, let me get started. I'm going to put on my Morgan Freeman voice here and try to, read through this. And shout out to my mother, Wendy Anderson. She edited this, and she edits most of my stuff that I write. Okay, so this blog post is called "A Look at STEM: What is ADME / drug metabolism." And also keep in mind, as you're reading this, as a Black man, there were no STEM professionals in my family, so I had no exposure to this growing up, and I did not get exposure to this until I got to the graduate school level. We didn't even talk about this at my HBCU, and so some of these things that are very much dependent upon who's in your personal ecosystem and just who's in your network. You might get exposure to these things early. My graduate advisor at the University of Michigan, his father was a scientist, so he got exposure to science early. And then some people get exposure to it later, and some people don't get exposure to it at all. So here we go. Black Guru's here as well. Thank you for coming, everyone. You could be somewhere else doing something else, but you chose to be here for a little while. Okay, so this reads: if you swallow a pill and it simply sits in your stomach and then passes through your feces, it technically wasn't absorbed into your body. Okay. Similar to the fields of pharmacology and toxicology, ADME / drug metabolism is a well-established field dating back to the 19th century, and it is very complex with respect to the wealth and depth of information available. It is still evolving today. The goal of this post is not to address every detail of the field, but instead to give readers a basic introductory understanding of the discipline. Further details about the many aspects of ADME / drug metabolism can be accessed online or in scientific journals. Okay, so with that, this is a vast field. There are researchers working on this. There are researchers doing this in academia. There are researchers working on this in drug companies. There are researchers working who have these aspects in the biotech companies, and this field is everywhere. So this one blog post cannot do justice to the entire drug metabolism world. So just keep that in mind. In my pharmacology and toxicology posts, and those are beneath this video if you want to read them, I briefly discussed pharmacokinetics, toxicogenetics, and the absorption, distribution, metabolism, and excretion of drugs and other xenobiotics. So those are compounds that come from outside your body. Endobiotics are created inside your body. These areas are collectively comprise the exciting field of drug metabolism. Whenever a new drug or industrial chemical is produced, several key aspects of the chemical must be characterized. How much of it gets absorbed into the body, where it goes in the body, and how long it stays there. The answers to these questions are collectively known as a molecule's ADME profile. And a tremendous amount of work goes into this type of research. It's very critical information as it helps characterize the chemical's subsequent pharmacological or toxicological effectiveness and properties. As you read through this post, keep a couple of key questions in mind. How much of the molecule gets absorbed into the body? Where does it go once absorbed? And how long does it stay in the body? Is it transformed into something new? And how does it leave the body? Okay, I'm going to stop and give a quick visual so you guys can see what I'm talking about. Let's see. So let's go to PowerPoint because I don't think I saved that as a JPEG. Stop screen share. I'm going to jump around a little bit during this one. Give me 1 second. Did I close it? Why don't I see it? Application window, your entire screen. Science. I don't want the training. All right, people, bear with me for a minute here because this happened yesterday or the day before. So close that. Let me reopen it. There it is. Okay. There it is. Okay, so what we're talking about here, just visually, is this. So we're literally talking about, so you have your pharmaceutical over here, or your drug of abuse. And this is the acronym here, ADME, and this is what it's referring to: absorption into the gastrointestinal tract here, distribution, where it actually goes in the body, metabolism of the molecule, and then excretion, which relates to your body's elimination of the molecule. So this is a visual of what I'm talking about in this blog post and in this live stream. Okay. And show. So let's go back to our guide here. Screen share. All right, so we're back. Before walking through the ADME acronym in greater detail, let's first talk about the 3 organs that impact a chemical's ADME profile the most: the liver, the kidneys, and the small intestine. As described later in this post, other organs can impact a chemical's ADME profile, but these are the 3 major players. I will try to explain these organs in the context of this post in the simplest way possible. So going back to some of Nicole's content, Nicole does gene editing, and I think she, if she hangs around for a little bit longer, she can come up and we can talk a little bit. But she looks at effects on the liver in the context of cancer and those kinds of things. But there are multiple contexts for liver function, and the argument could be made, well, for every organ in your body, that if you don't have that organ, you'll die. But proper liver function is very, very critical for how your body handles drugs. And the same is true for your kidneys. So going back to the opening quote, if you take a pill and it simply sits in your stomach for a brief period of time and then passes out through your feces, then it technically wasn't absorbed into your body, said the same professor from my pharmacology post, who distinguished the discipline of pharmacology and pharmacy. This anecdote described how drugs and man-made industrial chemicals, in the classic sense, must be absorbed into the bloodstream to actually have been absorbed into your body. And I'm going to show you a visual of that shortly. There are other ways a chemical can get absorbed into the body via inhalation or dermal exposure, for example. But for this post, I'm going to focus for the most part on oral absorption. And there's a picture there of your GI tract. I actually have this model boxed up in a room. I took it on career days when I was doing career days at schools here in DC. If absorbed into the small intestine, the molecule of interest then passes through the hepatic portal vein into the liver, where any number of things can happen to it. After leaving the liver and entering the general circulation, the molecule is for the most part cleared through the kidneys via the urine, but in some cases it can be deposited back into the GI tract and leave the body through the feces. Molecules can also be exhaled depending upon what they're transformed into once absorbed. Any molecule not absorbed by the small intestine leaves the body through the feces. Okay, I'm going to show another visual. I want to look at the chat for a second first, though. Okay, I still got 6 people here. So I'm going to stop this. Okay, so this, this one, this is a busy slide. I've used this in lectures before, but this is basically showing the 3 routes of absorption. So chemicals can enter your body either through the oral route, which is when you ingest something, you swallow it, your food, your classic pharmaceuticals. Inhalation, you can absorb a chemical into your body via the inhalation route, and then you can absorb a chemical dermally. A senior scientist in my division quoted these numbers once, and that's that the GI tract receives 21% of the cardiac output. The inhalation route receives 100% of the cardiac output. And your skin receives 9% of the cardiac output. So this is referring to the blood that your heart pumps out. Each of these routes of exposure receive different amounts of blood. So if you inhale something and it passes very easily from your lungs into your circulation, you can get a lot of a drug or a toxicant into your general circulation. But what I just referred to in terms of the hepatic portal vein and the small intestine, this picture kind of depicts that. So this little tube here represents your small intestine. Whenever you ingest something orally, that molecule or those food particles eventually make their way down into your small intestine. That if it's going to get into your body it has to be absorbed here into the wall of the small intestine, and it goes into the hepatic portal vein. And that portal vein runs right into the liver, where in this instance, a pharmaceutical, any number of things can happen to that pharmaceutical in the liver here before it goes out to the rest of your body. And I'm going to come back to that. These things here, I'm gonna just discuss this quickly and move on. But these here, these OATP, these OATPs, that stands for organic anion transporters. Those are drug transporters, and I'm going to come back to that. This is the bile duct. I'm going to talk about that shortly. And that's it. So let me go back to the blog post. Okay, it's got 8 in here now. Screen share. Okay, I can't emphasize enough the importance of the liver and kidneys, which both perform numerous critical functions in the body. For this particular context, the normal function of both are critical to the body's handling of both endogenous and exogenous chemicals, which is why physicians, nurses, and pharmacists inquire about their function early on when patients are admitted for care in clinical settings. With that in mind, I'll break down the ADME acronym and why these skill sets are so valuable for scientists who gain them. So let's start with absorption. As described earlier, the absorption aspect deals with how much of the chemical is absorbed into the body following oral ingestion, passage into the small intestine, the liver, and finally into the bloodstream. The properties of the chemical itself can dictate how much of it is absorbed, particularly its size, and for simplicity, whether or not it's charged or neutral. So molecules can either be charged or neutral, and chemists and pharmaceutical companies, they have to look at how the chemical is going to arrive in the small intestine, because that's going to dictate how it's going to get absorbed into the GI tract. As described in my pharmacology blog post, pharmaceutical companies designate molecules as either small or large, and many large molecules can only be administered via injection into the bloodstream. While there are several experiments that can help characterize a molecule's oral absorption, pharmacokinetic and biliary excretion studies are the most specific. Just briefly, by treating animals with a radio-labeled compound, the amount of compound can be determined by quantifying the amount of radioactivity in the blood, urine, and feces over time, telling scientists how quickly the molecule was absorbed in addition to the amount absorbed, and if these two metrics change with increasing dose. Okay, so I'm going to show some more visuals here. I don't want to overwhelm you guys with my reading. So let me, and I appreciate you all being here, listening to this. Okay, so we talked about absorption into the GI tract. So this is basically the GI tract here, and this is the actual small intestine here. And small molecules and large molecules. Let's unpack that for a second. Okay. I created this slide in one of my videos about 2 months back. Small molecules and large molecules, that's relating to the size of the molecule. And as you can see, I think these cutoffs vary, but a small molecule, I looked this up, is designated as any molecule 900 Daltons or grams per mole or less. So in this instance, we're looking at ibuprofen. That is 206.3 grams per mole. That's a measurement of its molecular weight. So this is considered a small molecule. A large molecule ranges from 3,000 to 150 Daltons or grams per mole. So this right here, this big guy is, this is Humira. Let me see if I can say this, adalimumab. So that's 144 grams per mole. So that's a large molecule. So that has to be injected directly into your bloodstream. You can't take that orally. Because of its size, but also there are enzymes called proteases in your digestive tract that would cleave this up before it even got absorbed into your bloodstream. So small molecules versus large molecules. Let's see. I don't see any questions there, so I'm going to go back and read some more. Where did I leave off at? Pharmacokinetics, biliary excretion. What is biliary excretion? Biliary excretion refers to this. And as you can see, this is all closely intertwined with physiology. So you have to know how the body works when you're doing pharmacology and toxicology, and obviously cancer biology like Nicole does. So this is another picture of your small intestine here. This is your molecule getting absorbed into the liver. And what you see here is that there's a bile duct going from the liver back to your small intestine. And so in some instances, depending upon the molecule, it can get absorbed by the portal vein into the liver, and it can circle back out into the small intestine. There's a thing called Entero hepatic circulation, which basically is this. What that means is that the molecule can get absorbed into the liver. As I'll show you, it can get modified in a certain way, and then it can go back out here into the small intestine, and those modifications can be completely wiped away by bacteria. I believe that's bacteria. So all these things impact the absorption of your molecule in your gut. Okay, almost done here. Okay, metabolism. Once in the body, molecules undergo biotransformation. That is, classes of proteins called enzymes can transform the structure of a given molecule by breaking it down into multiple pieces or adding on new functional groups, altering its properties so that it's more readily eliminated from the body. In some instances, this biotransformation turns the molecule into something toxic, which can cause injury to the liver or other tissues in the body. Excuse me. The metabolism aspect of ADME involves a separate discipline called enzymology, which focuses just on the enzymes themselves, their levels in cells or protein, their protein expression, their rates of their reactions, kinetics, their structures, etc. There are actually multiple classes of drug metabolizing enzymes, but the most prevalent class, at least as it relates to the liver, is the cytochrome P450s. Pharmaceutical companies pay particular attention to this class of enzyme and a host of others as they greatly impact the bioavailability of the drug. The first pass effect or first pass metabolism occurs when a drug is significantly metabolized before it gets into the general circulation due to metabolism by liver enzymes. Some of the clinical aspects of metabolism will be further discussed later on in this post. By the way, while cytochrome P450s are classically associated with the liver, we now know that they are expressed throughout the human body as well as all plants and animals. So let me show you a couple of pictures of that. Nameless is here. What's going on, Nameless? So cytochrome P450s and metabolism. So we're back to my PowerPoint presentation. Metabolism. So we typically think of metabolism as in two phases. There's phase 1 and there's phase 2. Phase 1 involves metabolism by an enzyme called a cytochrome P450, which is designated by this CYP3A here. And it's usually some form of an oxidation reaction. And so if those of you watching this have never seen this before, just keep in mind that this is a molecule here. This one is called sinobufogen. All these drugs have these exotic names. What you want to notice is that this reaction with this enzyme and this molecule, this parent molecule, is giving us two metabolites. So these are two variations of this parent here. If you look here, you can see that after this reaction, we have a hydroxyl group here on this molecule, an M1, and you have a hydroxyl group here on M2. So this enzyme modified this molecule here so that there was a hydroxyl group on it here and a hydroxyl group on it here. So there's a huge chemistry, there's a huge organic chemistry component to pharmacology, toxicology, and drug metabolism and ADME. Let me see what other points I described so I know what to show you guys. So that's phase 1 metabolism. Phase 2 usually involves, let me blow this up. Once that oxidation reaction has occurred, there can be a further step which involves glucuronidation, which is putting a sugar group on the molecule, sulfation, which involves putting a sulfur group on, or there could be some sort of rearrangement of the molecule. It can get tagged with something called glutathione, or if it gets activated, it can, in a toxic way, react with proteins and or nucleic acids like your DNA or your RNA. But that's metabolism. That's the metabolism portion. The first pass effect. Let me just talk about that really briefly before we go on. What the first pass effect refers to is that a doctor may give you a dosage of a drug, and it might need to get to another site in the body. So you might need the drug to get to the heart, or you might need the drug to get to the bone, or you might need the drug to get to you might need it to perform some sort of immune response. But the drug can be metabolized to an extent right here in the liver to the point where it never gets to its target because it's metabolized significantly. So that's called the first pass effect. So scientists and pharmaceutical companies and regulatory agencies have to keep that in mind when they're designing drugs. How do we get it through the liver? Does it naturally get through the liver? Or do we have to modify the molecule itself so that it gets through the liver in a very efficient way? So I'm going to read some more. Okay, so we're going to skip down to distribution. This one is simple. Thank you for the likes and the views. I appreciate this. Once in a body's general circulation, the molecule can travel to many tissues of the body and can accumulate there for short or long periods of time, depending upon the tissue and the properties of the molecule itself. Give me a second. I didn't share that with you guys. And this blog post is in the description box below, along with the others. Let me read that again. Once in the body's general circulation, the molecule can travel to many of the tissues of the body and accumulate there for short or long periods of time, depending upon the tissue and the properties of the molecule itself. If the molecule is particularly nonpolar, so if it's neutral, it can accumulate in fat tissues, or which is problematic in pregnant females, because if it's being stored in the fat, it can be released at some later point in time unknowingly. It can partition into breast milk. So depending upon the properties of the molecule, it can partition into breast milk, and it can go straight to the baby, a nursing offspring. Molecules can also bind reversibly to blood plasma, allowing for an increased internal dose. So sometimes the molecule gets into your circulation and it binds the albumin, which is a protein in your plasma. It can bind, and then you can get more absorbed. And then once that second fraction gets absorbed and eliminated, that molecule can come off your albumin and you can get a second dose. And I'm going to show you a picture of that distribution, so you could see it just again, what we're talking about here. Just bear with me for a second. Yeah, PowerPoint, we know. Okay, so let me go back to StreamYard. Let's see, brand. Where's overlay? Yeah, overlay. I see Black Uru left a comment. I'm going to read that in a second. There it is. When we talk about distribution, this is what we're talking about. Once that drug or that toxicant or that molecule gets absorbed into the general circulation, where does it go? Because once it gets into your bloodstream and the heart starts pumping it everywhere, it can go any number of places. It can go to your heart. It can go to the heart. It can go to your testes. It can go to your ovaries. It can go to your lungs. So this is what we're talking about when we talk about distribution. Where is the drug going, and is it accumulating? And if it's accumulating somewhere, how long is it hanging out in your body? That's very, very important. So let's go back. Let me look at the comment section. I see Black Uru left a comment. Let's see. Nameless says, I'm glad we could talk about it and introduce another means of influence to explain our condition/culture. At Black Uru Speaks, shout out to Nicole Ali as well. Black Uru says, I hope the manosphere embraces what you and Nicole are presenting in that area. Mastering that area may be a major key to our fixing many of our social and cultural problems. Agree. If anyone is new coming in here, please like the stream. Please subscribe to the channel. If you want to make a donation, my Cash App is in the description box below, but I'll leave it here. Yeah, piggybacking off of what Black Uru just said, keep in mind that, a lot of this in our community is not regularly talked about, and you have to ascend high in your education even to just start to have knowledge of this stuff. Let me go back to my screen share. Okay. Excretion. Let's talk about excretion. And as you can see here, there's a picture of your kidneys. These are two very, very important. We have two of them. You can actually live off of one, but your kidneys are very, very important. And once these malfunction and they're no longer usable, that's when you have to do dialysis, which is something I hope that I never have to experience in my life. Excretion refers to, excuse me. Excretion refers to how the molecule is eliminated from the body. Typically, the urine and the feces are measured to determine how the molecule is eliminated. Detection in the urine indicates that the molecule was absorbed to some degree into the bloodstream as the kidneys filter out aqueous waste from the blood. So that right there is why you never ever want to go on dialysis, which is why you have to take care of your diet and make sure you're fit, because your kidneys, their main job is to regulate fluid volume in your body, and also is to keep the things that you need and get rid of the wastes that you don't need. And once those break down, that's when you have to do something like kidney dialysis. But they will also, if those break down, that's also going to impact on how your body handles drugs and other compounds. Poor kidney function can lead to prolonged bioavailability and a subsequent toxicity, which is why clinicians always inquire about it as described earlier. Not all of the absorbed chemical exit the body through the urine, though. It turns out that absorbed chemicals can empty back out into the GI tract from the liver via the bile and then be eliminated through the feces. I already talked about that. Drug transport. This aspect doesn't traditionally fall under the ADME acronym, but it's an important field that is now being actively researched in academia and industry. It also deals with how cells may concentrate the chemical in tissues or remove the molecule from the target tissue before it can exert its function. So in the early 2000s, I remember that they were just, when I was a graduate student, they were just talking about drug transport. Companies were just thinking about this. It's 15 years later, so I'm certain that we have a much more extensive knowledge of what this is and how it works. Pharmacogenomics and toxicogenomics. These new and exciting fields look at the genetics unique to individuals to determine the best treatments and dosages for individuals. Genetic differences in levels in the drug metabolizing enzymes mentioned can result in drastically different effects of treatment with a given dose of a drug depending on the individual. The same is true for an individual's reaction to a toxicant. So we've talked about this recently here on this channel. And I'm working with another group, and we're going to talk about this again as well, to a larger degree. I don't see the, give me one second. There it is. Just give me 1 second, folks. Go to the overlay. So you've heard me and Nicole in our discussions about COVID and the coronavirus and hydroxychloroquine and chloroquine. You've heard us talk about. Let me hide that. Yeah, you've heard us talk about glucose 6-phosphate dehydrogenase. This is a genetic polymorphism that the COVID-19 pandemic has helped to unearth. This apparently is a very, very prevalent. The word is escaping me right now. This is a very, very prevalent condition. And there are whole groups now working on looking at what drugs exacerbate this condition here. And apparently African Americans, we have a lot of people who are deficient in this enzyme. So there's a question now whether or not COVID impacts this. And folks are concerned that there are certain drugs that exacerbate this. What it typically manifests as is hemolytic anemia. Which is represented here. So because the individual doesn't express glucose 6-phosphate dehydrogenase, they are more susceptible to hemolytic anemia from certain pharmaceuticals. And there's concern now that COVID-19 helps to exacerbate this condition. So there will be some more content on my channel regarding just this, but I think the point I want to get across is that we're not all genetically the same. And there are differences in our gene expressions, which impacts the expressions of enzymes, in this instance, drug metabolizing enzymes. And that means that basically we all handle drugs differently. And if you overexpress certain enzymes in the liver, that can limit the effectiveness of specific drugs. Let me take this down. And let's finish up here. Let's see. So why is all this important? Whether in a hospital setting, a pharmacy, or in the pharmaceutical industry, creating a new food additive, a pesticide, or a cosmetic, it's important to have as clear an understanding as possible of where the molecule goes in the body and what its fate is following ingestion. As described above, physicians, whether in general practice or in the emergency room, have to gauge a patient's liver and kidney function as those two organs will dictate how long the pharmaceutical stays in circulation, again, its bioavailability. Even a pharmaceutical designed to be therapeutically beneficial can be toxic if it remains in the body too long, if its levels exceed a certain dosage level, if it's transformed into something toxic, or if there is a drug-drug interaction. Drug-drug interactions are typically the result of one drug causing an increased internal dose of another drug due to inhibiting or preventing metabolism by the enzymes described in the metabolism bullet above. In graduate school, we learned about the classic case of terfenadine causing abnormal heart rhythms that could lead to death by increasing the amount of circulating erythromycin, which is an antibiotic. So I think terfenadine was an antihistamine and erythromycin is an antibiotic. Both of these drugs molecules normally work by non-cardiac mechanisms. Terfenadine was removed from the market once its ability to cause this deadly drug-drug interaction was recognized. Chemical, beverage, and pharmaceutical companies all have to know what happens to their molecules in the body for several reasons. A drug can be highly effective at preventing cancer cells from multiplying in a laboratory setting in dishes and flasks, but unless it is readily absorbed into the intestines and can actually get to its target tissue in the body as its untransformed structure, it's useless. In some instances, a drug can get to its site of action, but the cells of that tissue can adapt and effectively expel the molecule before it gets a chance to exert its function as described earlier. Pesticides, which are sprayed on agricultural commodities, often make it to our dinner tables in low levels where we do ingest them to some degree. So going back to what Nicole has talked about in terms of cancer, some cancer cells adapt to be able to immediately kick the drug out. So it might be a fully functional drug, it might be a quality drug in terms of efficacy, but that particular cell line might adapt and say, you know, we're going to kick this back out. Another very important context for ADME/drug metabolism is food safety, which is a key consideration for food and beverage companies like PepsiCo and Quaker Oats. As a matter of fact, at a family dinner earlier this year, a discussion about trisodium phosphate, a preservative used in Captain Crunch cereal, which had other industrial uses, caused a stir amongst my relatives. I had to remind them that both the ADME and toxicity profiles of this preservative had already likely been characterized and cleared through extensive studies by the company and the Food and Drug Administration. All right, folks, we're toward the end here. Let me see if there are any questions in the chat. Are any comments in the chat? No comments or questions. Okay. And I know this information, if you've never heard it before, is kind of foreign. So I appreciate you guys being here. We've discussed what ADME / drug metabolism is, but where do these scientists work and where do they receive their training? Okay, so this is important for the generations coming next. ADME / drug metabolism scientists work in pharmaceutical and chemical companies and in government and in the government performing regulatory functions. They receive their training for the most part in academic settings and labs specializing in pharmacology and toxicology, both of which have ADME / drug metabolism as a major component. There are some labs that strictly study one of the many aspects of ADME / drug metabolism, but they are in the minority of research groups in the biomedical sciences. So these people, the people with this training, those people are rare. Scientists with training in drug metabolism will almost never be without jobs. Again, this group is rare, said one of the professors in my graduate department at the University of Michigan. While we know that there is no job that is 100% secure, this particular professor was stressing that ADME / drug metabolism scientists are critical parts of most companies. The divisions of those companies responsible for these types of studies are typically titled drug metabolism and pharmacokinetics. So DMPK. If you go, to Pfizer or Merck or AbbVie or Biogen or GSK, they have a DMPK division, and that's the drug metabolism arm of the company, and that they need that arm, and that arm, no matter which drug target they're working on, they need that arm to get their drug registered. No matter what disease the company is interested in, it is essential that they understand the chemical's ADME / drug metabolism profile for their own purposes, and when submitting packages for approval by the Food and Drug Administration or other regulatory agencies. The same is true for the chemical companies. Depending on the degree level earned and where the scientist is employed, ADME / drug metabolism scientists can earn starting salaries of $60,000 to $70,000. That depends on the company, depends on your degree level, and it depends on which location you're working at. This could easily start at $100,000 as well. There are numerous scientific tools and technologies that ADME / drug metabolism scientists use, but I'll mention two of them briefly. The first is the mass spectrometer, also known as the mass spec. The accompanying picture shows a mass spec. Mass specs are not used solely in ADME / drug metabolism studies, but they're very important to the field because they can detect and identify molecules in whole blood, plasma, tissue samples, urine, and fecal samples at very, very low levels. So it's a sensitive instrument. More importantly, they can detect changes in the structure and the identity of molecules once they have gone through the body, and can help predict a drug / industrial chemicals efficacy or toxicity. I want to show this because I think it's important to keep in mind that when we talk about the biological sciences, there's a lot of tech involved. When you think about cancer and you think about pharmacology, toxicology, you think about drugs, you think about animals being treated, but a lot of the measurements, there's a lot of technology integrated into the generation of this data. So give me 1 second here, and I'm going to show you. It looks like my PowerPoint presentation went away. Let me do it me close that and open this back up. Okay. So this is a mass spectrometer. I did not, as a student, I did not get to use it because my graduate advisor bought a couple of these, but only certain people were, only certain people had projects that dictated them working on this and that required them to work with this instrument. But it's in essence a chemistry tool, and that's something my graduate advisor emphasized to me, because I was kind of salty, because I was thinking, okay, how come this is the instrument I need to use to get into a pharmaceutical company? Why can't I use it? Well, my project didn't dictate it. But essentially, what he communicated to me with it was that in using this instrument, it's a chemistry tool. So it's not just using the instrument, but you also have to know how to interpret the data. Well, let's look at how it works first. And again, I didn't use it, but I know enough to talk my way through this a little bit. So basically what happens is you have your sample here from your animal or from your in vitro experiment. It gets injected into this chamber here. You have an electron beam source. You have a heater that vaporizes the sample. And then so what this electron beam source does is it ionizes the contents of your sample, and it passes through this accelerator here. There's a magnet which separates these ions out, and you have a detector here which calculates basically the mass to charge ratio of your ions. And so with this, the scientists can tell what happened to their drug. They could tell what it was metabolized into. They could tell if it bound to a protein. They could tell what functional groups were added to it. That's why this is such a valuable instrument. And I wanted to show this to illustrate that when you're doing the biomedical sciences, you're not just working with animals and cells and proteins, but there's a lot of tech involved in it as well. Another tool used in pharmacology, in this instance, ADME/drug metabolism, is this. This is an HPLC, high performance liquid chromatography. That's what that acronym stands for. So again, this is there's tech involved in this. So this is the instrument here. This is probably a more modern model. You have a fluorescence detector. There's a column in here. There's a sample manager. You load your samples in here, and there's a solvent manager here. Basically, in a nutshell, this is another variation of it. You have your computer here, and this is your instrument, and you have your solvents up here. This is the chemistry tool, though not necessarily as sensitive as the mass spec. So what happens is, once you run your experiments with your animals or your cells or your in vitro system, whatever that is, you load those samples into your instrument here in these vials. And then over a period of time, this particular scientist has numerous samples. The instrument injects those samples onto this column. This is probably a reverse phase column, which means that it likes more neutral molecules. Those molecules adhere to the column, and then you subject it to something called reverse phase, where you expose that column to an organic solvent, and whatever's on that column comes off, and you get, I didn't add one to this slideshow, but you're not going to get this. This is from a mass spec, but you're going to get something similar based upon the chemical identity of your sample. So that's the HPLC and the mass spectrometer. Let me stop my, you know what? I don't think you guys even saw that. I'll throw it up afterwards. Let me finish reading. We're just about done. And I'll put the link in the chat if anyone else wants to come up and ask about anything I've read here. Okay. Technologies and methods are always changing and evolving, but the mass spec is currently a very important tool for ADME/drug metabolism. Currently, pharmacology and toxicology scientists and industry are moving towards decreasing animal usage and towards more in vitro and in silico methods, which are giving rise to the use of physiologically based pharmacokinetic models, where the fate of the molecules can be predicted using various constants and inputs and computational models. We're currently in the early era of these methods. So we're moving to a place where fewer animals are going to be used, and everything's going to be basically, a lot of this is going to be done on computer. I'm going to summarize this last paragraph by saying that most science disciplines have professional societies where scientists network, share their information, and exchange ideas, and also where people get hired. So for the drug metabolism world, the key societies are the American Society for Pharmacology and Experimental Therapeutics and the International Society for the Study of Xenobiotics. And if you talk to Nicole, there's a cancer professional society as well. I can't think of what their name is, but they have a meeting. And so each scientific discipline has its own world, and it has its own culture, and it has its own ecosystem and its own network. So these are the two societies for pharmacology and for drug metabolism. I'm going to stop talking. I'm going to put the link in the chat if anyone wants to come in and say a few words. That was a lot, especially if you're not familiar with these fields. But, I think part of my role is to have these discussions and start these discussions and get this information out there, especially for my own people. So if anyone wants to come up and say something or ask something, the link is right there. I'm going to find a picture. HPLC chromatogram. Give me 1 second here. Images. Boom. Okay, this is good. I thought I did this yesterday, but I ran out of time, as always. Let me see if anyone has clicked on the link. Black Guru. Black Guru, good morning.
Morning, Dr. Dunbar. How are you doing?
Good. Hey, Black Guru, I appreciate you being here. I know that was again, for people unfamiliar with this, that was probably a lot. But I thank you, curious individuals who stuck around and humored me a little bit. What say you?
Oh no, not at all. Not don't at all view it that way. I mean, I actually think what you've discussed, presented here, and I hope this isn't a one-time thing, is an area that Black people need to become much more acquainted with. And I think, because I think many of our problems are digestive in nature, not just what we consume, but when we consume, the volume of what we consume. I think so many of our problems, so many of cancers and obviously diabetes and hypertension, the very unfortunately the deadly trio or whatever that Black people confront, have a digestive and nutritional digestive and nutritional elements associated with them. So I think it's vital for us to become much more acquainted with the was it ADEM, or is that the correct
ADME, ADME.
ADME. Yeah, ADME. Pardon me. Yep. Yeah, I think we should, as a collective, if you will, of people, start to become much more much more acquainted with the things. I do have several questions, and I hope I'm not asking anything that may be outside or slightly outside of your expertise. But one question I have is I've often wondered if we're overprescribed certain medications, and by overprescribe I mean in quantity or dosage or intensity, because of digestive and particular absorption issues. Are there issues or problems regarding inconsistencies within our ADME that result in us being prescribed medications at a certain intensity to account for problems in those areas? So I guess that would be one question I have.
So my answer to that would be okay. So just in summary, you want to know is there are we overprescribed?
Do problems in the areas that you describe result in overprescription of certain medications or prescriptions at higher intensities or quantities as a result of problems with them being able to properly assimilate, I guess, lower intensities or quantities of medication?
Yeah, so I'm gonna attempt to answer this way. As I described, we know that there are genetic differences now between all of us. So the way my body handles a drug may be different than the way your body handles a drug versus the way Nameless's body handles a drug. We know that let's see. We know that if you have kidney issues, that's going to affect how your body handles a drug. And I'm trying to think of I know in the naturopathic space, your gut health. There's a part of this that involves your gut health as well. So your GI tract is an environment all in itself. I mean, there are beneficial bacteria that actually live within us. So my answer to that would be we're all different. And the doctor, they don't genotype us to see what our enzyme expression levels are. And I think that was something that they're trying to move to, is more individualized medicine. But there are differences between us and how we handle these things.
Yeah, indeed. And, I wonder, because, well, for example, drugs are, well, maybe because you're in the regulatory area, you would be familiar with this, but, when I've heard or read that, typically, medicine, the developments in medicine or medical research are sort of based on, I guess, the bell curve of what works best or what works optimally for a white male. I've heard or read something to the equivalent of that. Is that an actual valid issue or consideration? And if it is, then how does or should that inform how us non-Caucasian males view our consumption of medication and medical practices in general?
Well, I think, yeah, we're going back to individualized medicine. And I think at one point, at least they were trying to start to ask questions about how different groups, are there inherent genetic differences in different groups? And I think we know that there is. So I think there is a danger in not taking that into consideration for different people and different ethnic groups.
Well, the sad part for Black people in particular is because we are even well, obviously there's a fairly high percentage of non-African genetics amongst so many of us, but even within the population of our African genetics, even that is not necessarily uniform for most of us. It may be from assorted different tribes throughout Africa. So how do you profile, medication for that type of genetic background would appear to be quite a quandary, to say the least.
Yeah, you have to, we would have to uniformly genotype everybody to know and understand who's expressing more of this, who's expressing more of that. How will this person handle this drug? How will this person handle that drug? But yeah, I mean, there's so much variability for us, depending upon which part of Africa we came from, depending upon, how much mixing there was, in our bloodlines. Yeah, it would be damn near impossible to give one blanket treatment recommendation for all of us because we're all so different, even within the Black community.
Exactly. I noted in the chat, now I'll just repeat it here. I usually on pretty much a daily basis, I usually consume for essentially breakfast and lunch, the equivalent of like a 32-ounce smoothie, which is sort of a combination of, I guess, coconut or walnut or coconut milk or whatever, certain mix of protein, powdered protein, sorted fruit and vegetables, and I'll even toss, a couple of, multivitamins in that mix, right? So I'm thinking I'm just kind of loading up on all the potential nutritional necessity, so to speak. Are you aware of any particular issues with respect to that type of food or nutritional intake? I perhaps should study that kind of thing more. I just have kind of embraced that as a way of sort of really kind of control and keeping track of what I consume, particularly at a caloric level, and especially protein. Sure, I got the right amount of protein, that daily protein, that sort of thing. But I'm just wondering, are there any ADEM or ME issues associated with that particular dietary plan that I should maybe be a little more considerate of?
Black Uru, I don't know. So you make your smoothie? Are there vitamins in the smoothie? Or
yes. Well, like I say, I'll toss some like I say, sort of a high protein walnut milk protein mix, the powdered protein, sort of fruit and vegetable, could be broccoli, apples, blueberries, maybe some raspberries and pears or whatever, and maybe even like some broccoli or spinach in there, and a couple of multivitamins, for men, that type of thing, maybe one a day or whatever. That's generally what I'll consume for breakfast and lunch, essentially, because I usually just kind of consume it from the morning until the early afternoon. I mean, that's for most of the week. That's what I consume. I don't consume as much of anything else for breakfast and lunch for most of the week. And I have definitely witnessed or observed some actual improvements in energy and slimming, fat reduction and that sort of thing. So I've seen and witnessed some good results from that, but I'm just wondering, just kind of inspired by your presentation here, if there are some other issues associated with that I might want to be more considerate of, that sort of thing.
Based upon what you described, I haven't heard anything that that would give me pause. Most days I do a NutriBullet myself with some of the same ingredients you described.
Okay.
But
that's reassuring.
Yeah. unless you had any other digestive issues, then I wouldn't. Yeah, I think that's fine.
No, I mean, in fact, I think if anything, it's kind of helped to improve some consistency in that area as well. So that's also been a a delightful benefit, if you will.
Okay.
But yeah, no, like I say, I haven't observed any problems with it, but I'm just, like I say, since we're discussing about the, absorption and distribution and et cetera, I thought it might be worth kind of adding a personal touch to the discussion.
Okay. I just want to show everyone this. This is an example of an HPLC chromatogram. This is the data that you would get off of an HPLC, a high performance liquid chromatography experiment, versus this one. This is what you would see off of a mass spectrometer. So this one, I'm not going to talk a lot about this, but this one, it's you're looking at time on the x-axis, and each of those peaks reflects what we call the affinity for that, whatever that is, for the column. So, and these will come off when you increase the organic solvent. And these guys here for the mass spectrometer, you're looking at the relative abundance of a sample, and you're looking at ions, specifically the mass to charge ratio. And depending, so here you're looking at identifying the size of these ions, and for the HPLC, you're looking at the chemical, in this instance, the hydrophobicity, or the organic affinity of the molecules. But these are, I'm showing this to say that these are tools that they use, and I think it's important to understand. A lot of people don't understand how much tech is involved, even in the biological sciences. So I wanted to show that, and getting hired into a pharmaceutical company or a chemical company, or even understanding the data you're getting back in a regulatory agency, you have to understand the technology, what they did, how they did it, and why they did it. So there's a lot of tech involved, even in the biomedical sciences. ANC worker, thank you for showing up for as long as you're here. Black Uru, do you have any more questions or any thoughts on anything?
Oh, I'm sorry. I was on mute. No, I don't think I have any more questions right now. But like I said, I look forward to. Well, I guess one question I do have, particularly with respect to drug consumption, because you kind of opened the discussion with respect regarding there appear to be some apparent failure of full or proper assumption of a drug or drugs that were being consumed, if you recall.
So say it again. I said there was
someone consumed a drug, but most of it or part of it or whatever was in their stool, which means that it wasn't.
Yes.
Yeah. So I guess my question in that regard is it a good idea to, if you have pills, for example, is it a good idea to grind the pills into some?
Okay, so that's I think I see what you're asking. So I would say, because I have seen that before, but what happens is, depending upon the molecule and the properties of the molecule, it might need a coat to make it to its site of action. So give me 1 second, Black Uber. I want to show you something that will illustrate this. I'll use this one. This is a good one. So depending upon the properties of the drug, they might need to put a coat around it. If you look at this diagram, the blue represents your actual active ingredient. That's your drug of interest, and then you have basically 2 coats around it. And so if you were to put this active ingredient, if you were to swallow that, it probably wouldn't make it to its site of action.
Make it to the colon. Okay.
Because you have to think about there's a change in the chemistry of the environment. The stomach is highly acidic, and the small intestine is more basic. It's more alkali. So you have to deliver these blue contents to the small intestine without them getting destroyed or chopped up. So in that instance, cutting it up wouldn't do you any good.
Sure.
Now, depending upon the properties of the molecule, it just might not be well absorbed. So even if you chop it up into little bits that you can see, by value of its structure, by nature of its structure, by nature of the functional groups on it, by nature of its size, it just might not be well absorbed into the gut.
Sure.
Which is why something like your biologics, like Humira, that's a biologic by nature of its size, but also because it's an antibody. There are proteases in the gut that would chew it up before it gets to where it needs to go. You have to inject this. You can't take this orally. So.
Okay. That definitely helps to clarify things. I appreciate that.
Okay. I'm going to, so we got 5 people. We'll go for a few more minutes. Black Uru, do you have any? Oh, NC Worker's here. Okay. Let me see what he has to say. Hey, NC Worker, thanks for stopping through. Thanks for coming up.
Thank you for allowing me to be up. Thank you very much. And good afternoon, everyone.
Hey, how you do, NC Worker? It's always a pleasure, bro.
Likewise, gentlemen. So how much does the effect of medicine, whether it's coded or non-coded, or whether it's dye or not dye, has an effect on somebody's body that is maybe endomorphic or things of that nature?
So I think you're asking, can the coat have an effect on a person's their health?
Yes, like as far as like because I know I work in a retail pharmacy store.
Yes.
So I have quite a few people that come in and ask, do you have any dye-free, or, my wife's pregnant and she doesn't want to have dye with the baby, things of that nature. So I know in studies there have been some cases, hence why we have dye-free medicine, that the dye, I want to say either it's red 40, which is also commonly found in sodas and drinks, along with yellow number 5, I believe. So I don't remember them all, but there's things in those numbers and colors that have had effects on people. So I just wanted to know whether or not does that also have an effect as far as somebody's genetic makeup, as far as their body frame and things of that nature.
Well, yeah. So depending upon the levels of an enzyme being expressed, and whether or not that enzyme interacts with that particular dye or that particular ingredient in the drug coat, in theory, you could see something happen. You could see some interaction happen. Typically, the coats, I believe the coats are made up of something that's inert or something that's non-reactive. I think that the companies look for that. But also you have to think about the amount. There's a threshold over which a chemical will cause its effect, and depending upon what that threshold is, if it's below that number, you're probably not going to see anything. But to answer your question, in theory, if a person expresses enough of an enzyme and they get enough of a dose, in theory, you could see something toxic.
Okay. Thank you.
Mm-hmm. Okay. And I think I have a picture here somewhere that shows that. Give me 1 second. If you guys have another question, you can ask away. You can ask me if you want. Oh, there it is. Let me show you guys this. Yeah, so another consideration is a drug's therapeutic index. So there's a range in which the drug is effective, represented by the green. And then there's a range when it's not effective, which is the blue range. And then there's when it's toxic, which is the red. And obviously you want a dosage in the green range. And there are things that can cause the level to go into the red. It could be overdosing, or it could be your body's not clearing it fast enough or effectively. So that's the therapeutic index. You guys have anything else you want to ask before?
Not right now, Doc. Like I said before, I appreciate this information. I think it's very valuable. I hope other people in this environment observe more of these sorts of presentations, because, like I said, I think this is an area in particular for Black people regarding our health that we should be much more mindful of and more educated in. So I appreciate you doing this. I look forward to seeing more of it, and I look forward to other people, many more people, observing the valuable information you presented. Thanks.
Okay. Well, if you haven't watched on the live stream, I'm speaking to the general audience here. This will be available on playback. Please like, share, and subscribe. If you want to make a donation, my Cash App will be in the description box below. But going back to what you said just now, Black Uru, understanding this information is important for several reasons. I mean, I'm passionate about leaving a blueprint, for those coming after me. So we don't talk about these things enough around our dinner table, in my opinion. But also, based upon what we're going through now in society, and based upon the onslaught of information about drugs and about COVID, it's important to understand the science behind some of this stuff. Because
exactly.
if you don't I'm talking about for the layperson. If you don't understand,
well, so you can ask the right questions when you're being compelled to commit to these things. Yeah,
exactly. And so that you can decipher what's real and what's reasonable, whether I should be in fear of this or whether I should not be in fear of this. Information will free you from, in some instances, if some people want you to go a certain way in terms of your thought, having the right information will allow you to question that and say, you know what, no, that doesn't sound valid. Or I saw this study over here that said the contrary to what's being reported over here. So it's important to understand the science, even as a lay person. So, okay.
I just want to ask, so what is your take on the COVID vaccine? Do you think it'll eventually come to be, or do you think that it's still going to take some more time?
Well, obviously, they're trying to fast track it. but there's a balance between fast tracking it and getting it right and creating something that's not going to make people even more sick, because you can create a vaccine that will actually do more harm than good. So there's a danger there. So it could be that they're never able to generate an effective vaccine, and we're able to deal with this by other means, such as with HIV. We don't have an effective vaccine against HIV at this point, and it's been 30 years. So they may be able to create an effective one, and they may not be able to create an effective one. But there are scientists working on other means of treating it as well, just like they worked on alternative methods for treating HIV. I want to be careful about discussing the vaccines.
And I didn't mean to put you on the spot. I just want to know I guess your generalization of thought of process of how things are going about. That was all. I mean, I wasn't trying to say see, even this doctor, I wasn't going to do. I won't try to say or do anything like that.
Yeah, it's I don't mind talking about it. I think on my channel, I've created a couple of videos laying out the regulatory process and laying out the dangers of going too fast, and just laying out all the steps that are involved in bringing a vaccine to market. The only reason I'm concerned about talking about it, beyond that, is there are a lot of conspiracy theories associated with it. There's been something circulating around recently.
I was just about to ask you about that.
Yeah.
Because especially
we're going to talk in code a little bit here. So I don't know what to make of that. And it's been hypothesized that as soon as the new leadership is put in place, this whole thing will evaporate away anyway. So I don't know. Intellectually, I'm kind of on a whole pattern as far as that all goes.
There's someone, only for some people, be like, see, I told you he was right.
Yeah, I mean, I've heard that too. And boy, talking about the implications of such, God, I hope that is not true. And I mean, well, I don't know if the integrity of the country can withstand that.
Mm-hmm.
I think that would have a fractious effect if it became quite apparent to a significant number of people that is what has happened.
Mm-hmm.
And we know what that means by that. I don't think the country can survive that. And I think we're now in this weird place that if some very positive thing happens very soon, that may be the very conclusion that many people will draw.
Oh yeah. Yeah.
So it leaves you in this weird place where you want this problem to go away, but how are certain people going to respond to it in terms of what it means? These are strange times.
Very strange times. Very strange times.
But thank you for your time, Doc.
No problem.
I won't take any more of it. I hope you've gotten some out of my exchange with you in that regard, as much as I've obviously received the benefit of your expertise here and your generosity.
Yeah, no problem. I appreciate always being able to partake in your wisdom. I appreciate your support. And thank you for the Cash App, and thank you to Nicole for the Cash App as well.
Sure. Nope.
Yeah, so I'm gonna go ahead and shut down. This will be, again, if you're not watching this live, there'll be a playback. Please like the stream. Please share. Please subscribe. And I think we'll stop there. There'll be more of this, and this will be used to set up other discussions, because I think that again our community is kind of behind in the sciences, and little things like this can help catch us up, and that has an impact on. There are going to be so many careers opening up and so many employment opportunities and sectors spawned from the stems that, if you're not in on these discussions, then you're going to get left behind. So I'm going to stop here. Gentlemen, thank you for coming up.
Thank you for sharing your knowledge and wisdom.
Okay.
Thanks, Doctor.
Thank you. And everyone else, enjoy the rest of your Sunday. And as I always say, your attitude determines your altitude, and I will talk to you the next time. Bye-bye.